We will start with gene regulation today under the chapter Molecular Basis of Inheritance. That is the 10th lecture in this particular chapter. So to start with, what is gene regulation? Gene regulation is controlling the amount and time of formation of gene products according to the requirements of the cell. That is to say, all genes, the coding genes, they are responsible for production of proteins through the process of transcription and then translation. The protein that is produced, which has their assigned functions according to the need, requirement of the cell, the quantity, the quality, the time of formation, it varies from cell to cell. So the cell itself will, or the genes itself will control. They need to control, they need to regulate that at this particular amount of time, that much amount of the protein will be required. At other times, it is not required. So why unnecessarily that production? So to stop that production and restart whenever it is required. All such regulations, control of the release of products of the gene is, in a nutshell, called as gene regulation. The controlling the expression of the genes is called as gene regulation. Now, in eukaryotes and prokaryotes, the gene regulation differs to some extent. In prokaryotes, the regulation is usually simple, as because the structure itself is simple, whereas in case of eukaryotes, the regulation, these are more complex organisms or complex cells, so the gene regulation in eukaryotes can occur at different levels. That means the control may be done at different points. Let us see what are those points. So gene regulation in case of eukaryotes may be done at the transcriptional level. That is to say, the gene, when it is about to express, when the gene or the DNA, the fragment of DNA is about to express, then expression is always, as we know, in the form, first it is transcription. When transcription happens, DNA will undergo the transcription process with the help of enzymes, polymerase and other enzymes, and result in formation of RNA. But if suppose that protein is not required, the product is not required at that point of time by the cell, then the gene will not allow transcription to happen at that time. If transcription is blocked, transcription is not allowed to take place, then quite obviously there will be no proteins formed. So what is the cell doing? What are the genes doing? They are controlling the amount of, or they're regulating the amount or time of production of the protein by blocking or by stopping the transcription of that DNA. When required, whenever the cell will require, again they will restart and let transcription to happen, so the cell will get that protein. So if transcription can be blocked, then we can, or the cell will be able to regulate the amount or regulate the functioning of the gene. This is first. If suppose transcription has already taken place, but cell does not require the protein, so what can be done, or what the cell will do? They might stop the processing level. Processing level, as we are familiar, the RNA, whenever it is formed, it is in the form of hnRNA, which later on, with the removal of the introns, non-coding regions, and the capping and tailing process, the mRNA or messenger RNA is formed. So even if suppose the transcription took place, gene could not control at this level. So what will the cell do? The cell might stop the processing level, because unless and until the processing of hnRNA is done, it will not become functional. It will not be able to produce the proteins. So the cell can control the gene, regulate the gene even at the processing level. Third, let us assume even after transcription, processing also took place. But where are these two events happening? Both these two events take place inside the nucleus. Now, for translation to happen or the protein to be produced, it is very important that the RNA is released out to the cytoplasm. So, once it is released out to the cytoplasm, only then translation will happen. So, what the cell or the genes might act in a manner that the transport level is not allowed. The transportation of the processed RNA is not allowed. Transportation of the processed messenger RNA is not allowed into the cytoplasm. The transportation takes place through the nuclear pores, which will be aided by other structures present there. Now, what will happen? They will block the passage. They will not allow the transportation of RNA. Unless and until RNA reaches the cytoplasm, in case of eukaryotes, they will not be able to undergo translation and produce the proteins. So it can be regulated even at the transport level. But if, let us say, transcription, processing, even transportation took place, but that point of time the gene does not require or the cell does not require the protein, then how will it control? It will stop at the transcriptional level. The cell will not allow transcription of that particular RNA transcript to undergo. It will not allow translation to undergo. It will stop the translational process by blocking. Different mechanisms will be there to stop that. So it will not allow translation to happen. So in eukaryotes, the cells can adapt any one of these four methods in order to regulate or control the expression of a gene to produce a particular protein. Now, in details, we will have to learn at the transcriptional level. But of course, we will not go in eukaryotes. We will not talk about eukaryotes. We will talk about prokaryotes, how the genes can be regulated in prokaryotes by controlling at the transcriptional level. Let us see that. Now, first, before going into the deeper concepts, we have to know what is operon. The operon concept was for the first time put forward in 1961 by Jacob and Monod. Now, what is operon? Operon is a unit of prokaryotic gene expression with regulated structural genes and control elements which are recognized by regulatory gene products. The words may seem a little complicated, but otherwise it's a very simple concept. It is a unit of prokaryotic gene. Genes, as we have already seen, the transcriptional unit. Transcriptional unit is made up of three genes, what we have seen: promoter, structural, and terminator. Other than that, again, some other genes are attached, preceded. They're attached along with it, which will together control the entire functioning or transcription followed by translation of those structural genes within that fragment. That all the genes together, it is known as operon system. The components of an operon are structural genes. As we all know, structural genes are those genes which are actually or which will actually undergo transcription, which is always preceded by a promoter. Now, in between the structural and promoter region, there is one more gene fragment, one more few sequences of DNA, which we call as operator. Preceding that again is the regulator gene. So these four, they will be a part, or these are the genes that will form the part of the operon gene system. Operator genes. These are also called as operator genes, which are lined up. Regulator followed by promoter followed by operator and then the structural gene. Now, inducer is something which can start the expression of structural gene or which can stop the expression of structural gene. So, inducer is something which is provided from the external medium. Not necessarily it has to be produced by the gene complex itself. It is something which will be provided from the external medium. Now, of course, in details, we will learn about it now with the help of an example. Lactose operon or Lacoperon system. Lactose operon or Lacoperon in short is an example of an operon system in prokaryotic cells. This is an example taken from the bacteria Escherichia coli. Escherichia coli is a bacteria, is a prokaryotic bacteria for whom the food material is lactose. They take lactose from the external medium, which is a disaccharide. Lactose is a disaccharide made up of glucose and galactose. So they take it from the external medium, digest it. Digest means break it down to glucose and galactose so that it can absorb to yield energy, so that it gets energy. Now, the Escherichia coli, when it has sufficient energy, then it need not break down lactose anymore. If it has sufficient reserve of energy, then it need not take up lactose and break down the process, break it down into its units. Breaking down of lactose, behind the breaking down of lactose, there is involvement of enzymes. Now, if the breaking down of lactose is not required in the cell, then why should the cell produce those enzymes that will help in, or that helps in breaking down of lactose? At that point of time, not required. Breaking down, not required. Digestion, not required. So why unnecessarily to produce the enzymes? So what will a cell do? Cell will not produce those enzymes because it is no more, there is no call for it. So what will happen? There will be no production of the enzyme. Now, no production of the enzyme means something is done at the genetic level, at the gene level. That is regulation. Now, whenever lactose will be provided, what will happen? The enzymes will again be formed. So that is how it is regulated. Let us see that in details. It is also called as inducible operant because presence of a compound will restart the system. Now here we have a figure. Okay, we have a figure of the genes, of the total genes that is making up the operator system or operon in case of here lactose operon. It is an example as I told you. Okay, so here these are the series of genes. This is the gene which will, the regulator gene I, we call it the gene I, which will produce the repressor. It will produce a protein called repressor. That is followed by promoter region. Promoter region of the gene is followed by a small portion called operator region, and finally we get the structural genes. As we know, prokaryotes, they are polycistronic. So they are polycistronic, that is why here we see three genes. All of them will be participating in a particular metabolic pathway. We have already talked about the polycistronic monocistronic concept. So here, assuming you have understood that, so here we can say, or we say that the prokaryotes, they are polycistronic. So here we see three structural genes lined up, each of them producing three enzymes. Beta galactosidase by Z, by Y permease is produced, and A will produce beta galactosidase transacetylase. So three enzymes will be produced. These three enzymes are actually the ones that can break down lactose. When the cell requires lactose, when the cell requires, Escherichia coli requires energy, requires the food, these enzymes will be constantly produced. They will help in the digesting lactose. When digestion process is complete, cell has sufficient energy, no more lactose absorbed, then there will be shut down of the production of these enzymes. That is the regulatory mechanism, and that shutting down and opening up, or we can say switching off and switching on, is carried by mainly by this regulatory gene. Let us see the functions here in details. The units that makes up the Lacoperon, structural genes as given here, Z, Y and A. Z for enzyme galactosidase, Y for permease and A for acetylase. Then it is followed by the promoter region, to which the polymerase will bind so that these three parts will carry out transcription. So binding of polymerase takes place here, which we have already seen in the transcription mechanism. It is followed by an operator region, that means operator lies between promoter and the structural gene. Here at this point is responsible for binding of the repressor. Repressor is a protein. Okay, repressor is a protein which is produced by the regulatory gene. Here, Lac-I. It is produced, this repressor protein is produced with Lac-I, which can go and bind with the operator region such that the polymerase cannot proceed, such that it will block the operator region, such that the enzyme polymerase will not move forward and will not help in transcription and will not carry out transcription. But if the repressor cannot bind with operator, then what will happen? The polymerase will easily move and then result in production of the enzymes. So that is the basic mechanism or basic principle. By allowing repressor to bind, process will stop, switch off. By not allowing the repressor to bind, not allowing, I repeat, or preventing the repressor to bind with operator, the route is clear. So what will happen? It will be switch on mechanism. And what will control the action of this repressor, its binding or not binding, that will be controlled by lactose itself, which we will call as the inducer. In the previous slide I talked about inducer. So here the inducer will be lactose itself. Let us see how. Now here the Lacoperon. Absence of lactose. Absence of lactose, there is no lactose here. It is in the switched off mechanism. Switched off mechanism. So here we have the regulatory gene. Regulatory gene which is undergoing transcription and translation, normally producing a repressor protein. Here is the repressor protein. Now ahead of it lies the promoter region. Okay, lies the promoter region. Here this is the promoter region, which is followed by the operator region and the structural region. These are all genes, these are all DNA sequences. Now what will happen? The repressor, once produced, it will bind with the operator. Its structure is such, it can constantly, it will go bind, move out. The second will come, bind, move out. The third will come, bind, move out. So it's a continuously happening process. As it binds, the polymerase, which gets attached to start with the transcription process, it gets attached upon the promoter. It cannot proceed towards the structural part to carry out transcription. Why? Because the repressor is here. Repressor will block it. It will constantly keep on blocking it. So what happens? The polymerase cannot move, following which there is no transcription. Once this, and where is this happening? It is inside the E. coli cell. Once lactose enters into the cell, what will happen? Once lactose has entered into the cell, here we see lactose, okay, which is made of glucose and galactose actually. So lactose enters into the cell and then it binds or it reacts with the repressor, whatever was available in the cytoplasm. It reacts with it such a manner that its configuration or the structure of repressor will change. It gets modified to such an extent that it can no more bind with the operator, which was previously very easily binding, as we saw here. It's a continuous process, as I told you. So one moves, binds. By the time the second one comes, the first will leave, the second will bind. Third will come, second one will leave. So it's happening. So suppose here the second had already got bound. By the time the third comes, the second has left, but the third one is unable to attach with the operator. Why? Because its configuration changed. The presence of lactose changed its configuration. It's no more able to attach with the operator. So what happens now? The operator route is clear here. The way is clear. So polymerase will start moving ahead. It will carry out transcription of all the three genes Z, Y and A, forming the enzymes. Enzymes are produced. What will the enzymes do? They will walk on lactose, break it down to glucose and galactose. So till the time lactose is present, what will happen? Transcription will continue till the time lactose is present. That is why we call lactose to be the inducer, because it has induced the system. So it will continue. Once all the lactose has been digested, broken down to glucose and galactose, no more lactose available inside the cell, then what will happen? The repressor will be free from lactose. So the next one, after all the lactose has been consumed, the next one we can go and block it. It will go back to this original one. It will go again to the switched off mechanism. And at that point, obviously, as there is no lactose, there is no requirement of this enzyme. So it's very much important that it remains blocked. Why? Unnecessarily to produce the enzymes. So again, it will go back to the absence of lactose, that is switched off mechanism, till the time it gets a lactose, till the time it gets more of lactose. So this is how genes are regulated. Here is the example of lactose breaking down mechanism, which we call as Lacoperon system in prokaryotic Escherichia coli cells. Same thing. Okay, again here it's in a diagrammatic manner. The diagram which we can show in pen and paper, we can actually draw it, a simple figure here. So promoter, regulatory gene which produces repressor. Repressor will bind with the operator region, preventing transcription. Absence of inducer, switched off. Inducer present. What is the inducer? Inducer is lactose. So lactose comes, it binds with the repressor. Here repressor was produced like the previous case, but this time it got bound and it can no more attach with the operator, thereby allowing polymerase to move and carry out transcription, followed by translation, which will lead to formation of the three enzymes, and that can help in actually breaking down of lactose to glucose and galactose. Once all the lactose is consumed, it will again go back to the switched off mechanism. So that is all about Lacoperon system. I hope you could follow the lecture. So next we will be moving over to the next topic of the series. The next topic, which will be on Human Genome Project. So in details we will be learning about Human Genome Project in our upcoming lectures. Thank you.